Hello, good morning everyone. I'm Dr. Harika Kishore. On behalf of Vista's Learning, I'm here as your biology educator. V-Learning, it's an online education platform which works with an ideology of providing quality education for all. Visit our website www.v-learning.in where you're going to get an access for all the live classes, recorded sessions, study materials and also concept videos. Moreover, you're going to get this lecture notes in the form of PDFs. And also make sure to get your acquaintance with all the channels available and subscribe to the channel Vista's Learning Class 11 and 12. If you find this session useful and interesting, make sure to click on the like and share this session with your friends. And also click on the bell icon so that you are going to get all the upcoming video notifications. Saying this, let's begin our today's session. So we have been started about the chapter and that is called as molecular basis of inheritance, which comes under the unit genetics and evolution. In the previous session itself, we got to know about the structural importance of the DNA, and by now we understand that DNA is going to be act as a genetic material in majority of the organisms, though RNA is also acting as a genetic material in some of the viruses where we already listed like HIV, even coronavirus, it is a RNA virus. And also we can have a discussion, we had a discussion about the importance of DNA structure. So in this session, let's focus on the important salient features of the helical structure that has been proposed by the Watson and Crick in the year 1953, right? So here is the double helical structure. Double helical structure which has been proposed by the Watson and Crick. So, coming to the salient important characteristic features of the double helical structure of DNA, it is made up of two polynucleotide chain. As we already have a discussion about how the monomeric units, what are the monomeric units when come to the nucleic acids? That are nucleotides, right? Now, these nucleotides combine together, linked together by means of phosphodiester bonds, and also one backbone of the strand, that is one single strand, have another single strand linked together by the means of hydrogen bondings. So, this is one strand and this is another strand. That means in a double helical structure of the DNA, we do find the presence of two polynucleotide chains. Whereas the backbone, if you come to the backbone, it is the arrangement of sugar phosphates. So if you look at this strand, we can find the presence of sugar and phosphate, again sugar, phosphate, sugar, phosphate, sugar. The same thing happens on the other polynucleotide chain. Again, sugar, phosphate, sugar, phosphate, sugar, phosphate. So here, the nitrogen bases, which are purines and pyrimidines. In the purines, adenine and guanine. In pyrimidines, what do you find? That is cytosine, uracil and thymine. We already know that uracil present in the RNA and thymine present in DNA. So here, this nitrogen bases backbone is constituted by sugar phosphate. Just now we have seen in the image. And the bases, here the bases are nitrogen bases. Nitrogen bases, also called as nitrogenous bases, which is of two types, purines and pyrimidines. And these are projected inside. See, this is a polynucleotide chain which projects its nitrogen base towards inside. Same thing happens with an opposite polynucleotide chain where this chain projects its nitrogen bases towards inside, like a ladder. So, the two polynucleotide chains have anti-parallel polarity. Anti-parallel polarity, it means one chain starts running from 5' to the 3', whereas the other chain from anti-parallel polarity, that is, it starts from 3 prime towards 5 prime. So if one strand runs from 5 prime to 3 prime, then see here, then the other 3 prime to 5 prime. So here like this, these are the two polynucleotide chains. These are the two chains of the double helical structure of the DNA, where we can say one runs from the 5 prime to the 3 prime direction and the other runs from the 3 prime to the 5 prime direction. So if you consider this, here is the free 5 prime phosphate moiety, that is what we consider this end as a 5 prime end of the polynucleotide. From here, it reaches 3 prime end where we find hydroxyl group. And if it is here, the hydroxyl group is present, OH group, that means 3 prime. And here is a 5 prime. That means each strand will run in an anti-parallel with respect to other strand. The two chains have anti-parallel polarity. That means if one chain has the polarity of 5 prime to 3 prime, the other chain will have the polarity of 3 prime to 5 prime. Simple. Now, here, coming to bases. We just now mentioned about the bases, nothing but nitrogen bases, which are again of purines and pyrimidines. Here, adenine, guanine. Here, cytosine, uracil, thymine. Uracil will be present in the RNA, thymine will be present in the DNA. So here, the bases in two strands, the bases in the two strands, these are the bases. This blue color line is a backbone, alternative arrangement of sugar phosphate. This one and this one. And in between, we find some stalk-like structures, steps of the ladder, and these are your nitrogen bases. Nitrogen bases. So here, adenine forms two hydrogen bonds with thymine that are present on the other polynucleotide chain and vice versa. Similarly, guanine is bonded with cytosine by forming 3 hydrogen bonds. As a result, always purines comes opposite to the pyrimidines. For example, if you consider these are the two strands of DNA, if we find purine here and we find pyrimidine on the other strand, vice versa. That means if you find pyrimidine here, it will be linked to the purine in the other polynucleotide chain. It may be like adenine and thymine here. So the same way here, like this. This generates uniform distance approximately every time we find what is the distance present at this will be approximately equally to the distance that is present throughout the length of the two polynucleotide chain. This generates approximately uniform distance between two strands of the helix. See, in this image, it is helically twisted. That is what we can see a narrow down here, and we can see the broadening of the two strands here. But actually what happens, that are placed equal at each and every point of throughout the length of the polynucleotide chain. Now, so here, as just now we mentioned that the two separate strands, the two polynucleotide chain twist together like this. It will twist such that it is coiled to form a double helical structure. That is what we call it as a DNA helix. The two chains, this is one chain and this is another chain, are coiled in a right-handed fashion. Even left-handed fashion is also there, but we find the right-handed arrangement, right-handed fashion. Twist or coiling is right-handed. Now, the pitch is here, one turn from here to here. One turn. See, this is a one turn. If here is the thing, one turn it takes, and here is a twist, coil happened, and from here to here we consider it as a one pitch, one turn. So the pitch of helix is, this is important, 3.4 nanometers. A nanometer unit is 1 billionth of meter. That is what? 10 to the power of minus 9 meter. That is 10 to the power of minus 9 meter. If you express in the units of nanometers, it goes like 3.4 nanometers. So the pitch of a helix is 3.4 nanometers. Now, and they are roughly 10 base pairs. What do you mean by base pair? Base pair is a pair of nucleotide. Here we have one nucleotide on the one strand, one chain, and another nucleotide will be present on the other chain. This will be joined together by means of the hydrogen bonds that are formed in between the nitrogenous bases. So here the pair of the nucleotide which has been attached by the hydrogen bonds, we call it as a base pair, one base pair. That is, one base pair is a pair of nucleotide, and here 10 base pairs. So approximately one, in this twist we find one base pair. 2, 3, 4, 5, 6, 7, 8, 9, 10. So all together in one pitch we do find the presence of 10 base pair in each turn. This is one turn, right? Now, subsequently, the distance between a base pair, the distance between one base pair and another base pair. So that is here. See, this is one turn, no? Yes. So here to here is one turn, right? The distance between one base pair to another base pair, this is one base pair and this is another base pair, and that is 0.34. One turn is 3.4, right? We know that each turn will have 10 base pairs divided by 10. That's it. The distance between one base pair to another base pair. Subsequently, the distance between a base pair in a helix is approximately divided by 10, that is 0.34 nanometers. Now, the plane of one base stops. See, this is one base pair and this is another base pair. Say for example, it is adenine, guanine, cytosine, guanine, cytosine, like this, right? So here, these are stocked, piled up like a books, one above the other, one above the other, with a distance, maintaining a distance of 0.34 nanometers, 34 nanometers, right? So the plane of it, this is the stocks, one above the other, one above the other, we do find the presence of the base pairs. Base pairs over the other in double helix. This in addition to the hydrogen bonds. In between we have hydrogen bonds. No, here we have hydrogen bonds. Hydrogen bonds. We are talking about this hydrogen bonds. Confers stability. It will give the stability to the helical structure of the DNA. Now, so here, so up to now, we understand that the helical is right-handed, and in a pitch, we can see 10 base pairs with each distance. That means the distance between each base pairs in a double helix is 0.34 0.34 nanometers, 0.34 nanometers. Now, the proposition of the double helical structure for DNA and its simplicity in explaining the genetic conclusions. See, by now we understand that the genetic material, that is a DNA in most of the organisms, will be acting as a factor where it will store the information which is needed to maintain the organism. And therefore, the understanding is always becomes revolutionary. It's keep on going better. Our understanding is going on better with the understanding of the helical structure of the DNA, which is going to act as a genetic material. Now, very soon, Francis Crick, we have come across this name along with the James Watson and Crick proposed the model of double helical structure to the DNA in the year 1953. And soon after this better understanding of the structure and coming to the conclusions, Francis Crick proposed the central dogma of molecular biology. What do you mean by the central dogma of molecular biology? That is, which gives that the genetic information, which says about the genetic information flows from the DNA to RNA and from RNA to protein. That means whatever the sequencing of the amino acids that has to become in the protein, that is decided by the DNA through RNA. So here, I'll write here. So this is all about the central dogma of molecular biology. DNA, which makes its own copy by means of replication, right? Now here, whatever the information which is present in the DNA are transcribed into mRNA, messenger RNA, and that process is called as transcription. Transcription. Now, the information which is carried by the mRNA, and it is useful to decide the sequence of the amino acids that has to be placed in the synthesis of protein, and that is translation. This is whole central dogma of molecular biology. That is what? DNA. Here, transcription forms mRNA, which is useful where the information is carrying to give the knowledge about how the sequencing of the amino acids has to be made in the formation of the proteins, that is protein synthesis. And this process here, we do find the translation, transcription, translation, right? Now, so here, as we already mentioned that in some of the viruses, we do find the RNA as a genetic material, but not the DNA. In that case, just now we mentioned that in the central dogma, the information which has been present in the DNA is first transferred to the mRNA and then to the protein. So here, as the RNA is acting as a genetic material in some of the viruses, like coronaviruses, HIV viruses, retrovirus, rhinovirus. So here, the flow of information is reverse because the information is stored by the genetic material, that is RNA, and it has to flow to the DNA. That means formation of DNA from RNA, that is a reverse transcription. See, if the DNA is giving its information to form mRNA to transcribe into mRNA, this process we call it as a transcription, right? Here, what is happening? mRNA, as it is a genetic material, the information is stored by the RNA because it is acting as a genetic material. Now, this RNA is going to convert into DNA, which is a reverse of this process. That means reverse transcription. So now, okay, let's now understand about packaging of DNA helix. See, up to now we understand the structure, double helical structure of the DNA. And the stability of the DNA is coming from the hydrogen bonds which are formed between the nitrogen bases. And the backbone is made by the sugar phosphate units, where we do find the phosphodiester linkages, right? This is all fine. Now, what is the need for the DNA to get packed? What is the need for this packaging? Now, packaging of helical DNA, that is DNA helix. Taken the distance between two consecutive base pairs, that means one base pair to the another base pair. What is the distance between the two base pairs? That we already know, 0.34 nanometers. If we express in the meters, that is 0.34 into 10 to the power of minus 9 meters. This is a consecutive distance which is present in between two successive base pairs. So here, if the length of DNA double helix in a typical mammalian cell, human cell, is calculated, simply we have to multiply with it, simply by multiplying the total number of base pairs. Well, we are coming across the numericals of the length of the DNA of some of the bacteriophages. In that session, we have also studied about the length of the DNA that is present in the haploid set of the human cell. And there, that is 3.3 into 10 to the power of 9. If the cell is considered, that is diploid condition, that is double to this. 6.6 into 10 to the power of 9 base pair. With the distance between two consecutive base pairs, we know that this is the diploid set, right? Here is the diploid set, base pairs, and now we know the distance between two consecutive base pairs, that is it comes around 6.6 into 10 to the power of 9 base pairs into 0.34 into 10 to the power of minus 9. This is expressed in the terms of meters, not nanometers, by base pairs. It comes around 2.244, that is 2.2 approximately, 2.2 meters. Now the question is, we started with why the DNA helix needs packing. See, this is a typical length of, what is the typical length? That is 2.2 meters is a typical length of DNA that is going to be present in a single cell. Where do you find this DNA to be present? The DNA will be present in the cell, particularly in the nucleus of a cell, right? Now, 2.2 meters, this is very lengthy. A length that is far greater than the dimensions of a typical nucleus. If you consider this is a cell, this is a nucleus. Inside the nucleus, it has to accommodate 2.2 meters of DNA inside it. How it is possible? That is possible because of packaging, very compact packaging. Approximately, the dimension goes like 10 to the power of minus 6 meters. In that small area, in that small space of the nucleus of the cell, the DNA has to get its accommodation. Now, how is such a long polymer packed in a cell? Another, we need packing because the length of the DNA is very, very long when compared to the dimensions of the nucleus. That is fine. But this is very huge difference. That packaging has to be very compact. In such a way, it is going to get its reside inside the nucleus of the cell. In prokaryotes are the primitive cells and eukaryotes are the advanced cells that we already know, such as Escherichia coli. Though they do not have a defined nucleus, we know that true nucleus will be present. That means a well-developed nucleus will be present in the eukaryotic cells, and pro means primitive, where in this type of cells we don't find the well-developed nucleus. Instead, we find the place where the genetic material is, that is DNA, is going to be present, that we consider as a nucleoid. So here the DNA is not scattered throughout the cell. Though, if you consider this is a prokaryotic cell, though the prokaryotic cell will do not have a defined nucleus, but the DNA will not be present scattering around throughout the cell. It is going to be concentratedly present at one location of the cell, and that part, what we call it as a nucleoid. DNA, what is the charge of the DNA? We know that is negative, negatively charged particle. DNA, negatively charged molecule. DNA being negatively charged, it will get held with the positively charged protein molecules, is held with some proteins that are having positive charge on them in a region, and that region of the cell we call it as a nucleoid. Here we find the presence of the DNA, and this DNA gets its association with a protein, and then it is packed in such a way that it concentrates in a region, and that region is called as nucleoid. Now, the DNA in nucleoid is organized. It is compactly present in large loop held by the proteins. This is an interaction of the DNA with the proteins, and in such a way that it will organize itself in the form of a large loop by associating itself with the proteins, because proteins are positively charged and the DNA is a negatively charged molecule. Now, this is all about the prokaryotic cells. Coming to eukaryotic cells, we know that eukaryotic cells are much advanced. They have well-defined nucleus in them. This organization is much more complex because the eukaryotic cells are itself shows the complexity in their organization when compared to the prokaryotic organization. There is a set of positively charged basic proteins, and that type of proteins are called as histone proteins. See, proteins are made up of amino acids. Amino acids are the building blocks of proteins. These are amino acids linked by the means of peptide chains to form protein. This is a protein. And the charge of the protein will depends upon the charge of the amino acids by which it is made of. If the basic amino acids which are having positive charge over it are present in the protein, then the protein will get the positive charge. That is the reason why adenine and arginine, which are having a positive side chain, as they are involved in the formation of these histones, the histone proteins have positive charge. The proteins acquires, they will get the charge. Charge depending upon the abundance of amino acids with charge side chain. If this charge side chains are negatively charged, then the charge of the protein will be negative because the abundant amino acids are negatively charged. More the negatively charged amino acids, then there is a more chances for the protein to get negative charge. If more number of the positively charged amino acids are present, then there is a more chance that the protein will get the positive charge over it. So here, histones are rich in basic, this is important, basic amino acids, that means positive charged amino acids. Residues, what are they? One is lysine, important to remember, and arginine. Both lysine and arginine have the positive side chains. Both the amino acid residues carry positive side chains. This is important. Just now we said that amino acids, if they have positive, then more abundant positive, we say that this protein also acquires a positive charge only. So, histones are organized to form a unit of octamer, 8 molecules, and that we call it as a histone octamer. So here, this is all about to understand how the packing of the DNA helix takes place. See, we have a long length of DNA. this will get associates with the histone proteins. How many histone proteins? 8 histone proteins. And here the negatively charged DNA, negatively charged DNA wrapped. See, for example, what we are telling, these histone molecules are useful to get associated with the DNA. This is the aptamer. I'll explain how it is becoming an aptamer. This is the image that you have got in your NCERT textbook. Here is a spherical structure which denotes the core of histone molecule. This is not single molecule. It is a combination of 8 molecules. I'll explain. 4 on this side. On the other side also we do have the 4 histone proteins. What are they? What are that histone proteins? H2A, H2B, H3 and H4. These proteins will be present in 2 sets. That means 2 H2A molecules, 2 H2B proteins, 2 H3 and 2 H4. So this is H2A, H2B, H3 and H4. The same set will be present on the other side. Like this. H2A, H2B, H3, H4. So 4 plus 4, 8. That means octamer. Here we have octamer arrangement. It is not a single molecule, but it is a core of histone molecules, plural, right? So the same 4 will be present on the next side also. That means other side also. Like this. And over this molecule, DNA, I'll use another color. DNA gets wrapped to become condensed form, like this. And the wrapping is 2 times per 1 octamer molecule. So look at here, positively charged histone octamer for here front side and other side opposite to, again we have 4. 4 plus 4, 8. That is what we are calling it as an octamer, right? Octamer to form a structure called nucleosome. And this structure we call it as a nucleosome. A typical nucleosome contains 200 base pairs of DNA helix, that is pair of nucleotides. Nucleosome constitute the repeating units of the structure in nucleus, and that is called as chromatin. So in a chromatin, we do find the interaction of repetitive units. What are that repetitive units? Nucleosome. What is nucleosome? Nucleosome contains octamer histone molecules, core of the histone molecules, as well as wrapped DNA around it, right? So now, and this will be seen like a thread-like stained means colored bodies, which will be seen in the nucleus. So this structure, see, this is one nucleosome, and here in this picture we have 2 nucleosomes. This is one nucleosome and this is another nucleosome. As just now I said that the DNA, as it is a very lengthy, it is too long in length, so it wraps around taking a support of the histone proteins, and these histone proteins forms an octamer. What do you mean by octamer? Octamer is a 8 histone proteins in which we do find 2 H2A, 2 H2B, 2 H3 and 2 H4. These are arranged such that the octamers will get wrapped by the DNA, and this will be, that means the DNA which is coming from one octamer and the DNA which reaches in another octamer, this DNA we call it as a linker DNA in which we can find the presence of another histone protein, that is H1 histone. So not only this set of histone proteins, we also do find the H1 histone protein in the linker DNA. So this is another nucleosome. These are the repetitive units of nucleosomes that do we find in the case of chromatin, and that chromatin will be seen like a thread-like structure. So if you look at an electron microscope, we find it as a thread-like structure. That means bead string model. So this will again get wrapped up and compactly present, which coils, recoils, and finally takes the shape of chromosome. So here in the chromosome, we do find the presence of more compactly, more coiled, recoiled structure of 2.2 length of double-stranded DNA that has been associated with the histone proteins. So now we came to understand how that much lengthy DNA is going to present in a very small dimension of nucleus of the cell. So this is how. Just now I explained, as it is given like a single molecule, it is not a single molecule, it's a core which consists of 8 histone proteins inside it, which is made up of H2A, 2 H2A molecules, 2 H2B molecules, 2 H3 histone molecules, and 2 H4 histone proteins, right? So here approximately it may go 200 base pairs. And this is considered as bead, and this is considered as string, thread. So here, the nucleosomes, if you look at under electron microscope, we do find like this. See, I will draw here. This line is like this. This is the DNA, right? Above which we have beads. And this view will get under electron microscope. The nucleosome in chromatin are seen like a beads, like you can imagine the pearls on a thread, string, like a string. If it is compared with the DNA helix, that pearls can be compared with the beads, that is nucleosomes. That what we call it is a beads on string structure, beads on string structure when viewed under electron microscope. And this beads on strings will further get coiled off, further get coiled very close to each other. Consider these are the beads. Beads are present here, and each bead, if you magnify, we are going to see an actomer structure. Same for backside, and DNA wraps around it 2 times. In between, link. Then again, it will link with another nucleosome, right? The beads on string structure in chromatin, these are the beads on string structure in chromatin is packed to form. It get packaged, compactly packaged to form chromatin. Fiber very coiled, very strongly, compactly coiled, that are further coiled and condensed. That condensed form we can see at the stage of metaphase. Well, we are discussing about the in the previous class, that is class 11, we come across a cell division where we have different phases like prophase, anaphase, metaphase, telophase. During the metaphase stage of cell division, we do find the presence of this chromatin in its condensed form. Now, the packaging of chromatin at higher levels requires an additional set of proteins. Along with the histone proteins, it requires an additional set of the proteins that collectively called as a non-histone. They are histone proteins, right? Some other they will need. And that other type of proteins, what we collectively call as a non-histone chromosomal protein. Non-histone chromosomal protein. Now, we have come across the histone proteins, aptamer. Now, in a typical nucleus, some regions some regions of chromatin are loosely packed and they are stained very lightly. That means light in color. And that regions, what we call it as a euchromatin. We can call this as a euchromatin, and this densely packed and dark in color region of the chromatin as a heterochromatin. Euchromatins will consisting of a more concentration of the genes. Therefore they are like transcriptically very active. Transcription will be actively done. Whereas the heterochromatin, we can see it is transcription happens in an inactive. That is, the stage is inactive. Let's understand. The chromatin that is more densely packed and which stains dark in color, that regions we call it as a heterochromatin. See, heterochromatin are densely packed, dark stained. Euchromatins are loosely packed, light stained. That are the difference between the euchromatin region and heterochromatin regions. Euchromatin is said to be transcriptionally very active, transcriptionally active, whereas transcriptionally inactive is heterochromatin region of the chromosome. Whereas heterochromatin is transcriptionally inactive. Transcriptionally inactive is heterochromatin. Transcriptionally active is euchromatin. The difference is like euchromatin is loosely packed, lightly stained, transcriptionally active. Coming to the heterochromatin, densely packed, dark in color, that is darkly stained, and transcriptionally inactive. That are the different regions which we do find in the nucleus. Yeah, that is it. So in this class, we have discussed about the salient features of the double helical structure of the DNA, as well as we come across with the model of nucleosome, how the aptamers are being involved in the high densely packing of the double helical structure of the DNA inside the nucleus of the cell, right? That is all about today's session. Catch you in the next session. Until then, keep learning with relearning. Thank you.